Yes, sir.
Okay. Yeah. Thank you, ma'am, for this introduction. I'm delighted here to present the on a topic like innovation in biotechnology shaping the future of sustainable agriculture. I also like to thank SRC College as well as Earth Sciences for this lovely initiative in STEM education. So before going deep into the plant science research, I'll quickly like to introduce Jintugo. Jintugo is a deep tech research innovation company in Bengaluru. So we work on CRISPR-based genome editing in plants to transform the agricultural sector for sustainable agriculture. And talking about the Earth Sciences, as Dr. Ramu has already mentioned about Earth Sciences, I quickly take you through Earth Sciences. It's a STEM education center where cultivating young minds are given a platform where they can do STEM research at the age of like very starting with the middle school and high school level. So it's a great opportunity for the students. They are doing chemistry and biology middle school classes, molecular biology and molecular chemistry classes for high school, also pre-med and surgical programs for high school students, and then the science fair, which are like local and international science fair students are attending. With the state-of-art facilities and equipments, this is a great facility for the young budding students to come and do such kind of research at that minimum, this kind of ages where it's a great opportunity for the students. So talking about this plant science research and innovation, I'll quickly take you through first the fascinating world of plant. So as we see in our surroundings, plant everywhere. So we see like their texture, color, and then also feel the taste and the scent, everything. But inside this, there is so many pathways, cellular, biochemical pathways going on inside the plant cell, because of which we can see the phenotypic characteristics in our surroundings. So if you see a plant, so leaf is obviously an organ of that plant. And then if you dissect this leaf, you can see the tissues, defined layer of tissues, and you can also see the cells. So if you do a dissection of a cell, you can see their defined organelles. And then the chloroplast, which is the major photosynthetic organelle, is regulating all the photosynthetic activities. So talking about plant cell, it's definitely a... Can someone just mute? I mean, making some noise. So plants. So talking about the plant cell, it's an intricate masterpiece where lots of biological activities are going on. And the plant cell actually does several activities where it is the building block of plants. It also has diverse cellular components, and also it regulates the dynamic cellular processes. So lots of biochemical and molecular level pathways are working together to give you lots of traits. So if you see a dissection of a plant cell, you can see this is the plant cell wall, and then you can see the cell membrane. So in plant cell, the cell wall is the measure which actually protects, it gives the strength and stability to the plant cell. And inside this, you can see lots of organelles are there, starting with the nucleus, which actually has this all the genes and genomes inside which everything is going on. And then you have chloroplast genome, which actually is involved in your photosynthesis activities. It has its own genome, which is a primitive genome. And you have also your mitochondria, where your energy activity are going on. Then you have Golgi apparatus, and you have endoplasmic reticulum, where ribosome actually lies and do the protein synthesis. So it's a dissection of a plant cell, just to give you an overview. And then if you see into the photosynthesis part, so as I told, chloroplast is a major organelle which does this photosynthesis. So the CO2 is actually captured in the chloroplast and by the enzymes, and then it is converted into sugar, and you also get oxygen released. and in this process water is converted into oxygen end of the day. So you get sugar produced, and this sugar is utilized by the plant for its own energy, which is also stored as a starch for its different organelle function. So what is the importance of photosynthesis? Photosynthesis actually gives us Earth's 80% oxygen. It also provides 90% food sources, and it's the largest biomass source in the Earth. So if you see inside a chloroplast, so it's a powerhouse of photosynthesis, which is a double membrane structure, and it also contains the thylakoid system where your energy is captured and then CO2 is converted into your sugar. And stroma are also the fluid filled cavities where actually this Kelvin cycle takes place. And the color, the chlorophyll, which actually gives us the green color of plant, is actually the green pigment which absorbs light, and where actually all this electromagnetic spectrum plays different role, and then it is actually helping in capture of light and also converting different CO2 is converted into your sugar. So all these processes are going on through chlorophyll as the major pigment which actually absorbs your main light source from sun. So if we talk about photosynthesis and its energy storage, as I already told, sunlight is captured and then it is converted into CO2 is converted into glucose, and that glucose is stored in different tissues in the form of a starch, which is later utilized by the plant for its own development. So there are three different kinds of plants, three different kinds of plants, like C3 plants, which are mostly rice, wheat, and cotton, and several other plant species. C3 is basically the 3-carbon compound, which actually helps the your carbon. So it's the first stable compound produced. So that's why it is called C3 plants. C4 plants are mostly the maize, sorghum, millet, all these very strength, which does not require much water to grow. So this kind of plants. So C4 again is the C4 compound is the first stable compound which actually is formed during the C4 cycle. So that's why it is called C4 plants. And finally, you have a chem plant where you have chem plant where plants like pineapple and cactus, which like mostly desert plants. So they generally capture your things in the night, and then it is converted during the day into sugar through photosynthesis. So these are the chem plants. So these are the three different types of plants. So now lots of work is already going on to convert rice into C4 plants, because it gives you lots of benefits, like you need less energy, you need less water, and then you can produce lots of biomass. And so it's a great opportunity to convert, if we can really convert C3 rice plant into C4 rice plants through genome editing. So talking about the future of photosynthesis research, obviously lots of opportunities are there for photosynthesis research, starting with enhanced efficiency. We can improve the efficiency of the plant. We can do bioengineering using genome editing and other GM opportunities where we can improve the photosynthetic parameters. So thereby, if there is a climate change, we can always, if you have a improved photosynthesis. So if you have a improved photosynthesis, so climate change condition you can somehow subsidize, and then it also helps in sustainable agricultural practices. So coming to genome organization in plant, I will be taking you through how genome is organized in the plant. So to discuss this, we will discuss about the nucleus, chromosome, and the genes. So again, I'll come back to this, and this is your nucleus, inside which all these activities are going on, the genomic level activities are going on. And if this was the nucleus, so inside this nucleus, you have the chromosome. So the chromatic structure actually has the helical form of DNA, which is entangled with histones at different point of time. So this is your, say for example, if it is your DNA, it has some coding DNA sequences which actually forms a functional gene. So these genes actually helps in your different processes and biochemical pathways for different activities. for plant development and other activities. So talking about the nucleus, it's obviously the main control center. It is surrounded by a nuclear envelope, and then it has a nucleolus and chromatin, which actually is the DNA and protein entanglement. So what DNA is? DNA is basically the building block. So you have your adenine, guanine, cytosine, and thymine. So these bases actually form the helical structures, and these are actually the your carry forward. These coding sequences are actually carry forwarding your generation after generation traits from one plant to another. So as I told, gene and gene expression are like gene is the blueprint of life. So when there is a gene expression, the expression is done through a promoter. So it is just on the 5' end of a gene through which gene expression happens. And from this gene expression, you finally get your protein produced, and that protein helps in different processes in plant. So this is the overall pyramid. If you see DNA, and then your transcription happens where your mRNA is formed, and then your translation where your protein is formed, and the protein is your final product, which actually gives you different traits and everything. So gene regulation, gene expression is regulated through different factors. Environmental factor is one major factor where like light, temperature, drought, several other parameters affect the gene expression. Also developmental stages regulate the gene expression. At different developmental stages of a plant, different genes are expressed. Some are like constitutive genes, which are expressed throughout the plant, throughout the developmental stages of plant, and some are like different stage-specific plants, stage-specific genes which are expressed during its different developmental stages. And also the internal signals are there, hormones and other signaling molecules which actually regulate gene expression, and lots of transcription factors which bind to the promoter sequences helps in regulating the gene expression. So far we have understood like how the plant cell works, and inside the different organelles, how they behave. And then now if I talk about the history of CRISPR-Cas9, so it was basically initially identified in bacteria. So these two ladies got Nobel Prize in 2020 in chemistry for discovering CRISPR-Cas9 genetic seizures. So it was, as I told, it was first discovered in bacteria. So this system is now like mimicked to use in eukaryotic system, where you have your target, this guide RNA, which is very specific to your genomic DNA sequence. And then once it binds, the Cas9 enzyme can detect. So this is your Cas9 enzyme. So it can detect wherever your guide RNA has bound, and after that it will just cut. So it will make double-stranded break, because of which your DNA is broken into two parts. Now there are several other ways by which you can, in the cell itself, this joining happens. So once this break happens, there is two process. One is called non-homologous end joining, where you don't need a template to join this; it is automatically joined. And then you have a homologous template repair method, where you provide a small template, and it can just repair this whole thing. So this is the idea now used like highly in both plant and mammalian system for genome editing purposes. So how CRISPR works is like you design a guide RNA. As I told, you design a very specific sequence, which is near about 20 base pair, and then you have your Cas9 enzyme, which once it get the guide RNA bound to your target sequence, can bind to the DNA and then clips it. So this is the whole process. Once it is clipped, then there is your repair mechanism system, which actually is present in your cell.